Hi everyone, I'm going to present our paper, Structured Causal 3D Reconstruction. This is joint work between Max Planck Institute for Intelligent Systems, University of Cambridge, and Milan. Our motivation starts from the observation that image formation is actually a physics-based process. Rendering is therefore an approximation to such a process which involves the causal interaction of multiple factors. But current rendering-based reconstruction pipelines did not take causality into consideration. Instead, they treat the factors as independent ones. Specifically, the typical pipelines take an image as an input and independently encode them into latent features. These latent features are separately fed into decoders and then to differentiable renderers to produce the reconstructed image. Our structured causal reconstruction pipeline has an additional layer of causal ordering. Specifically, with the features produced by the independent encoders, we use the adjacency matrix of the underlying causal DAG to neatly combine them into another set of features, which are then fed into the independent decoders. And our purpose is to answer the following question: is causal modeling really helpful in 3D reconstruction? To simplify our problem, we're only considering the following four factors: viewpoint, depth, lighting, and albedo. And we evaluate all possible dense causal orderings. And our answer to the question is that yes, causal ordering matters. In the figure below, we show the validation matrix on all causal orderings, and it is clear that some causal orderings are better than some of the other dense causal orderings. We present a different view from representing conditional distributions. Conventional 3D reconstruction pipeline decompose the joint distribution of the latent factors into a multiplication of the distributions of independent vectors, while our pipeline decompose the joint distribution into a causal chain. We'll also prove that under some assumptions, we can have better functional approximation with a proper ordering. How to learn a dense causal ordering? We can use a Bayesian optimization pipeline to solve for the following objective on unweighted DAGs. Specifically, for any DAG, we can train a model for a fixed number of iterations and compute the validation metric using the loss wave shown below. And then we feed the validation score into the Bayesian optimization pipeline to produce the best DAG. We can also learn a generic causal ordering, which basically means that the causal ordering does not have to be a causal chain. Here, the settings are simpler because we can relax the DAGs to be unweighted graph. And by relaxing the DAG to be continuous entities, we can actually unroll the inner optimization steps shown in the last slide to a few gradient descent steps. And we use meta-learning method to optimize for the outer optimization loop, basically the DAG parameters. We then add a regularization term to constrain the causal graph to be a DAG. On top of the learn dense ordering from Bayesian optimization, we can learn a generic causal ordering. And because we already have the dense causal ordering, we are guaranteed that the learned generic causal ordering is a DAG, so that we no longer have to add the regularization term. Finally, we can learn a dynamic causal ordering, which is dependent on the input images. And the adjacency matrix of the dynamic DAG is actually computed by the attention on the latent features by computing the pairwise inner products. And we simply optimize the following loss without any validation dataset. Here we show our results on the BFM datasets, and it's clear that our methods are better than the baselines. We show two different settings because we observe that for different CUDA versions, we have different numerical precisions. And we show that our methods converges faster than the independent structure causal reconstruction pipeline. And we show qualitative results on the selective phase dataset and the CARS dataset. Finally, we visualize the dynamic causal orderings learned from the dataset. On the left, we show the mean DAG learned across the whole dataset. And on the right, we show that the residual DAGs depending on each individual input images, and you can clearly see that they are different. Thank you.